This is TWIS. This Week in Science, episode number 736, recorded on Wednesday, August 28th, 2019. Hot Nudi science! Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with sleep, airports, and puzzles. But first, TWIS is supported by listeners like you. We thank you for your support. We really couldn't do it without you.
Disclaimer. It started a long time ago in a galaxy much closer than you might think. The one you are in and on the outskirts of. In fact, it was just a spark, or maybe it was a muddy blob at the bottom of an ocean. A simple interaction of the right combination of chemicals. However it began, life has come a long way since then, with domains and kingdoms, phylums and classes, families and genuses, species of all sorts. Life diversified in a brilliantly mind-blowing explosion of possibilities. So much so abundantly so, that it may be easy to forget that it is truly unique compared to what we have seen thus far of the rest of the universe. Out of all of those possibilities, out of the seemingly endless diversity of living things on Earth, you happen to be one of them, and a human being thing at that. Sarah, with a keen brain and a modest ability to communicate. And of all the places you could have been in this amazing world full of life, you chose to be here. And we are so happy you are, because you are in for another episode of This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to fill your heads with all the science news that we thought was really interesting this week. We found all sorts of fun stuff, as we love to do. This week, while there are fires burning in the Amazon and, we're hoping that people will jump to the rescue there, we've got science that we can talk about in the meantime. Find ways to find hope in this earth of ours. I have stories about sleep, right? Sleep's good, gives me hope for the future, our planet, and computers. And we also have an interview this evening with scientist and science communicator Dr. Richelle Tanner coming up in just a few moments. Justin, what do you have for us tonight?
So far, I have a Toxoplasma Gondii story, and that's about it. But there might be something else by the end of the show.
Something else. You might pick something else up. Okay, plenty of time to put something together. Just you go search for your stories right now. All right, Blair, what is in the animal corner?
Oh, my goodness. I have deaf birds, I have smart birds, and I have migrations.
Migrations. I love migrations. Mm-hmm. Migrations. Going places we like to go. Okay, well, let's get on with the show. That's what I like to do. It's a migration from the beginning to the end of the science stories. As we jump in, I want to remind everyone that you can subscribe to the TWIS podcast. If you have not yet done so, you can find us all places that podcasts are found. The normal places: iTunes, Google Play Podcast Portal, and on Stitcher, Spreaker, Spotify, Pandora, Radio.com, TuneIn. Oh my goodness, so many things. You could also find us on YouTube. We're on Facebook. Look for This Week in Science. But to start, to find information, TWIS.org is one of the easiest things to remember. Now, let's jump into this interview. I would love to introduce our guest tonight, Dr. Richelle Tanner. She is a climate ecophysiologist at Washington State University. She communicates about science and climate change and is a member of the Governing Council of the National Network for Ocean and Climate Change Interpretation, or NOCI, and that is nothing to do with Italian potato pasta. Welcome to the show, Dr. Tanner.
Thanks for having me.
You're welcome. We're so glad to have you on. I know that we've met briefly within the last year, and Blair works with you through the Noki network. But it's about time we brought you in to the TWIS show so that we could talk with you and about the work that you do, so that our audience can know more about you. So, to get started, what is a climate ecophysiologist?
Well, I like to break it down by all the parts of the word. So ecophysiologist just is ecology and physiology shoved together as one word, because we can't be bothered to say more than one, I guess. And it's basically looking at how the environment is changing how the body works. It's pretty simple. And when you add in climate change, it's just kind of understanding how changes in our environment that are human induced can change how the body works in lots of animals and plants.
And what do you specifically study with relation to this field of study?
Well, in terms of animals, I think you guys know that I work on nudibranchs and sea hares, but right now I work on mussels, which are the kind you eat, the kind you find in the supermarket. So broadly, all of those animals are mollusks.
Right, all in the family, the mollusca family.
Right.
Why are they interesting to look at? Why did you go from the nudibranchs and the sea hares to mollusks? What is the thing that ties them all together?
Well, I like to think of them as kind of the superheroes of our seas. So a lot of them live in these really extreme environments that we think are just having these really big shifts in climate, even in our current climate. So because they can deal with these really huge shifts in environmental fluctuations like temperature and how salty the water is, we think that they can help us understand how we will see a future in our environment and how we can start to adapt.
For us, I mean, adaptation, I'm just going to go to the store and buy some raincoats for where I live up here in Portland. Other people might work on their air conditioning, but animals have to adapt in different ways. So when we're thinking about it in terms to bring it to the point of climate change, what are we really concerned about with these creatures live in the ocean, and the water gets warm and it gets cooler, but it's pretty moderated. I mean, when we think of the ocean and we think of that moderate temperature, that doesn't change too much.
Yeah, so that is something that is kind of a common misconception. The open ocean is actually really maybe moderate compared to the things that we experience on land. But when you think of organisms that live near the shore, which most of the things that I study do, they live at this intersection between land and sea. And so not only do they experience those really cold temperatures if you live near the Pacific Ocean, they also experience the really hot air temperatures. And so they're going from like 40 degrees Fahrenheit all the way up to over 100 degrees Fahrenheit in the span of a few hours every day.
That's like an astronaut on the moon going from like night to day on the moon. They're going hundreds of degrees. But so what allows these animals to be able to withstand such temperature differences over the course of a day?
There's actually a lot of different ways that animals deal with this. The ones that can move even just a little bit by shifting their posture, they can do behavioral regulation of their temperature. These nudibranchs can't do that. They move incredibly slow. So all they have to protect them is their cellular response. So what's happening on below the levels of organization that you can see on the surface. And so what they have is these things called heat shock proteins that go around in their cells and find proteins that have been mangled by the effects of temperature, and they bring them into the heat shock protein and they refold them and then spit them back out. And so if you have lots of heat shock proteins, you can withstand more high temperature effects.
And we have these proteins too. So this is something that we have in common with these organisms. But I'm really finding it really striking how you mentioned the difference between the behavioral modification. So like, I can go inside into air conditioning or go under a shaded area. But there's this cellular adaptation that takes place, this ability that the cells themselves. And I mean, I'm just thinking of this as these animals that are cellularly manipulating themselves. It's this organismal response. It's like if you had a dish of cells of bacteria that had to respond, it's pretty much a similar state.
Mm-hmm. Yeah. And I think the important thing to remember there is that they're not always expressing these proteins that do all of this really important stuff. Because if you think about it, if you had to, generate lots of really expensive functions, like if you were always running all the time, breathing really hard when you didn't have to be, it's probably not great. And so they have this thing called plasticity where they're able to change their expression based on what they experience in their environment, either right then or a couple days, hours, weeks before, to try and, cut down on the costs that their body has producing these proteins.
Wait, weeks before?
Yeah.
So they also, are they predicting the weather? Is this what we should, instead of the 6 o'clock news for the week's weather, we should be cutting open mollusks to see
Ask
what
a
the protein
nudibranch?
content? Yeah.
So they kind of have this understanding of regular fluctuations in our environment, right? Just like we know what seasons are, and we're in summer right now, and we know that it's going to cool down. Based on how long their lifespan is, they can do the same thing. And that's why climate change, it can be really important to understand how climate change is going to affect them, because we're going to have different kinds of shifts in our future that they are not used to seeing. So their predictions of the past and the future are going to be wrong.
And so how did their responses impact kind of other animals in the intertidal, or how can our studying them help inform what we know about other species?
So some nudibranchs can be keystone species, which mean that they play a really important role in our ecosystem, whether that be they die when there's an El Nino event, which they actually do the opposite of that, or something really important eats them. That's kind of what a keystone species would be. So in that respect, we can kind of use them as an indicator of how well we're doing for everyone else in the food chain. But on a more biology theory level, understanding how the heat shock proteins work just across all of these species can give us a lot of information on what we can look for resilience in other species.
Yeah, it's like looking for clues. We know there's relation. We have these proteins in common, and we have these little threads. So are there things, clues that we can use to find out who's going to do well and who's not? I'd love to, you recently published a paper on nudibranchs and their plasticity and ability to withstand these changes in temperature. Can you tell us a little bit about what you found and what its implications are?
So I guess I'll preface this by saying that this project was not something that we really set out to do. It was just kind of a fun weekend exploration of the coast, and we saw all of these nudibranchs popping up. And so we decided to look at which ones were doing the best. And to understand why we were seeing so many. And one of the ones that popped up in the news everywhere, I don't know if you remember, I think it was in 2016, the Hopkins rose nudibranch, which was this bright pink, puffy pom-pom looking thing. And everyone wanted to know why it was there. And we actually found that that species has a really high tolerance for temperature. They can get up to 95 degrees Fahrenheit for a pretty substantial amount of time and still recover from that. But the really interesting thing that we saw was that when you raise the average temperature of their environment, so if you simulate kind of what climate change will look like, they drastically lose this ability. So they have a trade-off between how well they're able to manipulate their upper critical limit, so how hot they can get, and their plasticity. And so this paper was kind of a really great example of this principle in biology called the trade-off hypothesis, which is exactly that. There's a trade-off between the upper limit and how well you can manipulate that upper limit.
And so this was kind of surprising, right? Like you would expect that the individuals that are used to hotter temperatures would do better in a climate change scenario, right?
Right. And so we're thinking that it might have to do with some sort of energy trade-off. So as a physiologist, we always think about how we use our energy, how it gets partitioned in different aspects of our physiology, right? You could try and use all of it to run as fast as you can, or you can try and use all of it to run as long as you can. And that's kind of the same principle that's happening here.
I'm really fascinated also by this trade-off and what it means for which species are going to do well and which are not. And, in the ecology world, there's also these trade-offs between generalists and specialists. And there's the concern that specialists will not do as well in climate change because, say, their food species will disappear or particular aspects of their niche will not be available to them any longer. And so in thinking about this trade-off with the tolerances with how plastic they are versus how high they can go. You also found this in another study that was published with mussels as well. So you're not just finding it in this one species of nudibranch, you're finding this kind of across different species within this whole family.
Right. And it's actually outside of this family too. The whole theoretical framework was proposed by my PhD advisor in 2002 in crabs, porcelain crabs. And so it's really being seen in a lot of different species, but we're also seeing the opposite in a lot of species, like in diving beetles. If they have higher tolerance, they also have higher plasticity. But the thinking behind the trade-off hypothesis is that it all has to do with energy, right? And so you don't have an unlimited amount of energy to just keep increasing your tolerance and your plasticity. It has to stop somewhere. And so we're actually writing a paper right now. It should be in press in the next few weeks on melding that idea of the specialist generalists and the trade-off hypothesis, kind of thinking about an overall framework for energy allocation under these like really variable climate scenarios.
And when we're thinking about the energy that they're expending to tolerate these higher temperatures, what is that going into? They don't have to sweat. So, what is the actual energy expenditure that's occurring? Do we know that?
Well, making proteins takes energy, and transcribing RNA, which makes the proteins, takes energy. It all does. And so being able to mount a response and do it quickly is really energetically expensive.
And then there's also got to be sort of a trade-off. If you're making this protein, you're not making something else.
Right.
Yeah.
Or you're making less of it.
You're using basically the same machinery and pathways to get these proteins out, which means something else isn't getting expressed as much. And did you need it? And if you did, and now, but you're just having to fight the heat. That's a whole other series of trade-offs that are going on.
Yeah, a really tangible example of that is that these organisms that are constantly stressed and expressing all of these stress proteins, they aren't able to reproduce. And so that kind of limits the population success in the future.
Wow. So this is just all feeding back. It's like, hey, I'm more tolerant and I can survive a little bit more. But if this change happens more, then I'm going to die. But even beyond that, I'm not having any kids either.
No, right. There's no time. I'm too busy being successful at not dying.
Right.
You have to stop and reproduce.
Yeah, which kind of leads me to my next question, which is a lot of these animals, like mussels, are pretty sessile. They can't really move around, right? The nudibranchs, a lot of these guys are in tide pools. Really hard to get from one tide pool to another. So what kind of hope can we have? What expectation can we have for adaptation or kind of movement for these species to be able to make it through this really rapid change?
So I'll take mussels as an example. It's a little bit easier because they're broadcast spawners. So that means that their babies don't live necessarily where they live. And so they put out all of their gametes into the open ocean where it's not very stressful. And so you have lots and lots of babies out there, and then they come back to settle, and they come back along the entire coast. And so there's a lot of chance, they call it like sweepstakes recruitment. There's a lot of chances for them to make it to a place where they can survive, and in a different range. And in terms of having hope about this, even though we're finding these trade-offs, they are still well above where we expect to see climate change affecting these populations. And so we kind of have to walk the line between making sure that our audience understands that there's urgency in this problem, but also knowing that there are species that are going to be okay.
And in terms of that, all of these studies, what you're finding is the goal. It's not just to understand it scientifically, but also be able to make recommendations to field ecologists, to conservationists, to people who are working in, either conservation or in the climate change or ocean environments.
Yeah, I have gotten really interested in restoration ecology in the past few years, and that's kind of the idea of using what we have in the current biodiversity, knowing which ones are going to be successful, and kind of building our ecosystems around those. And yeah, I think that's a major thing that we strive for, but it can be really difficult to link physiology because it can be so abstract, especially at the gene and protein level, to management strategies. And so we're just trying to still work on that multidisciplinary link.
Yeah, you're going from gene and protein to individual organisms and then populations, and so it's drawing that line between all of those things to be able to get to the point where you can have strategies for restoration, right?
Yeah.
But it's all about reducing stress, right? So like, I think about
so many things in life.
Well, and I mean, stress, not like, oh my God, I'm so stressed out, but like, in stress factors, right? Because you were talking about trade-offs, and in climate change all the time, we talk about individual animals or ecosystems or populations that are responding to one very slight change in climate or in pH of the ocean, for example, because there's all these other things happening to these populations. So if we can reduce one stressor in the form of giving them more space or shelter, then potentially, right, there's an opportunity that they can respond better to climate changes.
Yeah. And I think this is kind of even deeper in the weeds, but what's happening in some restoration fields is thinking about how we can kind of help existing populations by bringing in individuals from other populations that are more resilient. They have this gene expression that we know is successful under future conditions. So they're actually doing this in some coral reefs in Indonesia and the Great Barrier Reef. They're starting to look at how we can bring in resilient corals from other areas to kind of help the corals in that area broaden their genetic diversity to, deal with a bleaching event.
So it's invasive species with a purpose.
They call it managed relocation. It is a very, very hot topic, highly debated whether that is useful.
Yeah, you got to watch the I word, right? Invasive.
Yes.
A dirty word.
We take animals all over the place sometimes because we want them to be there so that we can, raise them and eat them. Sometimes because we want them to outcompete some pest species that we don't want to be there anymore. And now because we want them to help out ecosystems at the genetic level, which I find fascinating. That this is getting to that whole thing of like, well, it's all... In the end, it kind of all comes down to genes anyway. And if you put the species together and they can breed, then maybe that's a good idea.
Yeah, some of these, they're not even moving species. It's just populations that they're moving around. Some people are all about using CRISPR instead and actually editing the genes. But I think we are quite a far ways away from that right now.
I think we're very, very far away from that right now. But I probably have read sci-fi stories about this, entire worlds that have been genetically created.
That would be so much work. Just take life that's from another planet and see if you could set it down somewhere that's kind of similar, and they'll find a niche eventually. Right? It doesn't have to engineer it that well.
You would think so, yeah. So, this gets to the kind of, we are always, as humans, manipulating our environment. We are the master engineers, aside from beavers, maybe? I don't know. But we are in the process. We have been manipulating the planet, and it has caused climate change. We have anthropogenic climate change ongoing right now. People are coming up with engineering solutions for that as well. But, where do you fall in the discussion of, what steps we should be taking when you look at the populations that you're studying and you're seeing the threat to particular ecosystems, to populations? Where does that put you when you start looking at the planet and what we have done to it?
Well, I think a lot of people think that scientists shouldn't have a view on this because it's sometimes seen as political. But I think we just have a responsibility to manage the world that we live in. So, a lot of the things that I study are going to be okay. And resilience is a huge theme of my research. And so in that way, I like to inject a lot of hope into my messages. And hopefully, along with that hope, I can encourage people who are either reading my papers or listening to my outreach talks that we need to take action as a community. And it's really not that hard. So I try and lead by example, but it can be difficult when we're going to conferences around the world. And so scientists are trying to look at carbon zero conferences, which is a really interesting new idea on how we would meet as a community and also uphold these values that we hold in terms of combating climate change. And then we also all just try and be a little more cognizant of how our research is impacting our community. And we want to make sure that we bring everyone in so that everyone understands their role in the whole situation and not just us telling them that things are going to be bad. And so that's kind of the approach I take, even though there are definitely more active ones that I could be taking, right? Like promoting civic action, all of these great things that Noki does. It's not something that I feel is where my best effort is spent.
So I kind of wanted to ask about that too, because listeners to the show that have heard me wax on and on about Noki before know how I'm connected, and it's a no-brainer because I work in environmental education and conservation education. Of course, I'm going to care about climate change communication strategies. So one of the reasons I have loved working with you in the Noki network is that you are this kind of voice for the research side of things and the scientific community, which are a really important part of Noki. But I'm just kind of curious how you became interested and now how you're using social strategies for climate communication in your work. Since, as you said, a lot of that is focused on kind of, the research side of things. How do you use that?
Yeah. So how did I find Noki and why am I still using it? Yeah. So I think being a scientist, first and foremost, is you don't have any impact unless you're able to communicate your work. And I think that's been lost recently, or not recently, really just like throughout science becoming sequestered in this ivory tower. And so I really wanted to understand how I could communicate my science, especially because I work in climate science, knowing and from experience, knowing that people are kind of hostile about that topic, how I could be approaching it in a better way to kind of have a more productive and honestly an easier discussion in a lot of my outreach talks. And the way that I use it right now is kind of more on the meta scale, I don't talk a lot to the public. I don't do this sort of thing very often, but I do come into contact with a lot of scientists who are talking to the public or who have the same level of engagement that I do. And as a bigger community of scientists, we can have a really big impact. And so if I can bring more of my community into the Noki community, that's what I consider a success in my involvement.
Nice.
Yeah. It's kind of having that trickle down effect of the information coming from you and potentially leading out to other people who will then have the information lead to other people, and the network grows. It's I guess a hub and spoke kind of model.
Yeah.
Awesome. We've touched on the heavy climate change, and we've touched on your science. Now, I would love to know, what is your favorite mollusk? What got you into the mollusks in the first place? Your sea hares. Tell us about your passion for that stuff.
So I saw my first nudibranch in high school, and it was one of the ugliest nudibranchs out there. It's called the shaggy mouse nudibranch because it looks like a dead rat when it's out of the water, like a wet dead rat. But for some reason, immediately I really wanted to study these animals. And I did my undergrad, not really in biology, but still knew that I was going to go to grad school and try and think about nudibranchs. And when I found my advisor at Berkeley, we kind of came up with this idea about sea hares because there were lots of them in his lab and no one was studying them. And I was really passionate about climate change already because my undergrad was in environmental studies, environmental policy, influencing our nation's decisions around our oceans. And so I kind of brought these two things together, and that's why I study mollusks and climate change. But it really started with that one that looks like a dead rat.
Awesome.
I really like dead rats. Hey, no. But it's not about the dead rat. It's about the organism. It's about the ideas. So you're working now in this as a postdoc in this mollusk lab working on mussels. Do you foresee continuing to look at this particular area of animals, or do you want to branch out? I mean, what are you looking at in the future?
Wow, it's like you're asking me what the contents of my job application due next week are.
There we go.
I think what's important for me in the future is to keep looking at patterns of environmental variation and how that will change with climate change. In terms of what animal I'll study, it does not have to be what I'm currently studying. I'll always love nudibranchs. They'll always be that weekend side project, just like they were in my PhD. They weren't my PhD either. But I hope to stay with aquatic systems, but it's really, looking for the best organism that will answer that question about climate change is what I'm really interested in.
Right. And I think what you're mentioning, the side projects, and also how this paper on nudibranchs was a weekend side project. It was this, hey, I'm going to go out and, look at this. That's fascinating.
And our best paper yet.
Yeah. Yeah, I'd love to hear your thoughts on kind of how that weekend side project for you, how it fits into your schedule and how it keeps things going. And, just for the scientific process, there are lots of times when you think of things being very planned, observation, hypothesis, you plan the experiment and do all that stuff. But a lot of science also is it's kind of this magical moment of discovery.
Yeah, and I think that's what keeps a lot of us engaged, is having these little things that we have this closeted interest in, or something that your boss tells you is don't do that, focus on your main project. But everyone has them. My side projects right now are finishing my papers for my PhD, which is a little bit less interesting.
Right.
But when we went to the beach to go look for them that first time, it was me and one of my best friends from grad school. And we were just going because we loved looking at them. And then we realized that we had a large enough sample size to do an experiment that we had coincidentally already set up in the lab. And we thought, this will be easy. And here we are 3 years later, now sequencing these animals with another collaborator spending thousands of dollars, and it's kind of snowballed, but in a great way.
Yeah, talk a little bit about that. I'd love to know. So that you're sequencing them now, and you're actually going to be digging into what's changing.
So we're looking at the population genetics. We have a collaborator at Berkeley. So both me and Eric, the first author on the paper, have moved on. He's in France and I'm in Washington. But someone that we knew pretty well, Rory Bowie in the Museum of Vertebrate Zoology, is pretty interested in population genetics of these animals. And we actually don't know anything about their population genetics. So this would kind of be a first pass at looking at the genetic diversity, how their physiology might map back onto that genetic diversity, which is something that we've done with the sea hares already. And so it's kind of the next step into looking at possible mechanisms, which is always the end goal in these theoretical biology realms.
Right. It's the, physiology. You want to actually get to the mechanisms, what's happening in there.
Why?
How does it work?
Yeah.
It's the how question, not the why question.
Yeah, which is so much harder.
Exactly. Blair, did you have any more questions about nudibranchs or other aspects of Dr. Tanner's work?
I have a million. We could talk about it forever, but I think that ultimately, my question is kind of what in all of these kind of different experiences you've had with the intertidal to this point, with looking at these different organisms and their responses to climate change. What is your big takeaway thus far? Have you had a big aha moment? Are you seeing this big trend that you really want people to take away from your work so far? Is there something, this big question mark that you have that you're still trying to find out? Because it seems like there's this through line between all these different things you've been looking at very clearly, right? So have you seen a link? Is there some big question or thought that's coming out of all this?
Well, I think the first very small one is that everything is going to be okay. The ecosystem will move on with or without us. So it's just a matter of whether we want to kind of slow the changes that we've already made. But the big question that's still remaining for me is, again, this mechanism. What underlies this ability to be plastic, this ability to change your physiology with certain cues in the environment, and especially what Justin mentioned like weeks back, the predictability of the environment. I'm really interested in the heritability of these short-term changes, because it can't happen at the genetic level. It's happening within one individual's lifespan. So how are they doing this, and how are they passing this ability to their offspring? I have no idea.
Yeah, I'd love to. There's a lot that also has been coming out in plant research related to epigenetics and these effects that are multi-generational, where you have one parent that has stress, 3 generations down the line, they're still exhibiting some aspect of this stress that was experienced during their life. So it's not genetic. It's something different.
That's probably what my lab will be looking at once I move past here. We are working on some preliminary stuff right now, but it's hard when there's no genome.
Right. Yeah. Right. I mean, you have to have, what is the basic blueprint that all of those mutations and the epigenetic manipulations work on? What are they actually building on?
And then that doesn't matter. And you're going to want a ton of RNA-seq data to see what's being expressed at specific time points along the. There's more fun ahead.
Oh, then you want protein, because RNA isn't actually what is being produced.
Proteins. I'm sorry. Yes.
No. You want both. That's exactly what I'm doing right now. We have all of it. That's exactly what I'm doing.
Do you know, is the nudibranch genome a very large one? Do you have any idea?
Yes, there is a model sea hare that's used for neuroscience research, actually, the Aplysia californica. And, oh my God, it's 800 kilobase. It's not the largest and not the smallest. It's pretty average sized.
All right.
Yeah.
Nice.
It was an animal with an average sized genome. But outsized responses. Yes. I love getting to hear about all this stuff. Where can people find out more about you and follow you online?
I have a website that I try and keep updated when I'm not doing research. It's justmyname.com, RichelleTanner.com. And all of my social media handles are exactly the same. Just my name.
You got in there early. There are either that or there aren't
no,
a lot
no
of
one
people
has
with
my
your
name.
Right. So Richelle, RICHELLE, Tanner, TANNER.com. You can find her website, which has a blog, information on her research projects. And like you heard here, you can follow her on social media as well and find out more about Noki and her science and science communication. I'm not going to say escapades.
I like that, actually. I sign on to escapades. Yeah.
It's hard to say.
And I'll also, just because we were talking about Noki a bunch, I'll throw in there as well that you can find out more about Noki at climateinterpreter.org. At Twitter, it's @_Noki, NNOCCI, or on Facebook, National Network for Ocean and Climate Change Interpretation. Because we did talk a little inside baseball a few times about it. So if you heard something you're really interested about, you can hop on over there. There it is.
And you could read more about the study there too, because we wrote about it there.
Yeah, Richelle wrote a blog all about it. So yeah.
Oh, and something I did see for those scientists who might be listening to this and wondering, oh, what is this methodology of talking about science that Noki uses? What is their framework for talking about stuff? You can actually learn about it for free from Noki. There is, Richelle, do you have the information on that? I don't have it right offhand, but I read about it as I was surfing through web links today.
We have multiple courses in lots of different formats. They're all outlined on Climate Interpreter. You just have to make a free account. We also can bring a training to you or your institution, and you just request one of those. You can email me, because the email will eventually come to me anyway, or request it through Climate Interpreter. But yeah, we'd be happy to give a talk at your conference or institution.
Yeah, and Kiki just pulled up as well. There is a Frameworks Academy that is an online course. I want to say it's 10 hours, something like that. Mm-hmm. It is a multi-module course that gives you kind of a nice overview to the methodology that is used. But like Richelle mentioned, we are all about person to person. So we as a community are all over the country and are super excited to come to your organization or do local trainings for people. So yeah, absolutely reach out to Richelle about that.
I think this is wonderful. Richelle, thank you so much for joining us tonight. It's been wonderful to talk with you. And I know you have a pup to go put to bed. So
it's not enough. Richelle has a full-time job. She's looking at nudibranchs on weekends. She also has a puppy. So,
and it's not a nudibranch puppy. It's a real dog puppy.
Yes. Left the pet nudibranchs behind in grad school. Thank you for having me.
Yeah.
Thank you for being here.
Yeah, thank you so much for joining us. I hope our paths cross again. It's just wonderful to see the work that you're doing, and thank you for sharing tonight.
Thank you.
Thanks for stopping by. So fun.
All right, everyone, just to remind you, RichelleTanner.com is the website you can use if you are interested in more in the work that she's doing. And Noki, we will have these links on our website, TWIS.org. It is time for us now to take a quick break. So stay tuned for more This Week in Science. We will be back with all that news about sleep and airplanes and whatever it is that Justin brought. So that's just surprise science coming up after the break. We'll see you soon. everyone. Oh, yeah. This is This Week in Science. Thank you so much for listening to the show or watching it if you're watching it on YouTube right now. I would love to remind you at this moment that if you haven't paid a visit to TWIS.org, that is where you can find a pop-up window where you can sign up for our newsletter. That's right. We're sending out a newsletter now. Not on a monthly schedule. You can get our newsletter if you sign up. If you don't get that pop-up, you can email me, Kirsten, at thisweekinscience.com, and I will put you on our mailing list. Also at twist.org are our wonderful links to all sorts of fun, twisty things. If you're interested in helping This Week in Science keep doing what we do, twist.org, you can find links to all sorts of ways that you can help support the show and support independent, woman-run science podcasting. That's right, independent. Nobody else runs this show. We do. You do. You help produce this show. We cannot do it without your help. The Zazzle store is a place where you can find goodies that have all of the TWIS logos and also art from Blair's Animal Corner calendars. There's lots of products, t-shirts and mugs and wonderful things that would be a lot of fun for you to have that do support what we do at TWIS. Also, the Patreon link that'll take you to our Patreon community where you can help support what we do through a monthly donation. $10 a month, and we will thank you on the show. That's right, $10 and more a month. We will thank you on the show. $25 a month, you get a t-shirt, all sorts of fun goodies for you over at our Patreon community, plus some freebies that only patrons of TWIS get. You can subscribe here. Click on the orange button to subscribe. YouTube, iTunes, Google Play. It'll take you to an easy subscription interface. So if you know somebody who is not subscribed, send them to twist.org and tell them to click that subscribe button. Also, an easy way to make a one-time donation is to click the yellow donate button on the sidebar. That is a PayPal button. It will go to our PayPal account and help us to buy Blair a new computer. We are going halvesies with Blair on a new computer, which she needs because her computer's dying. It's pretty much dead. We're testing out a Chromebook tonight, and we're not super happy with it. So we would like to have a computer that can handle the streaming and do all the nice things. And so to do that, it's going to cost us some money. And, well, it's going to cost Blair some money, but it's going to be for a lot of TWIS work. So we are going halvesies. And so we would love your help to be able to make that happen. We would love to get a new computer, keep the show going. And I don't know, we might be having to buy Justin a new sound card also. So there's all these things happening at once. Whee! Technology. Okay. But anyway, your help will help us deal with these technological issues and, with style and aplomb, and to continue bringing you This Week in Science weekly to your ears, to your eyes, to your brains. We cannot do this without you. We cannot do this without you. Thank you so much for your support.
Explain things you've heard more than intuition.
And we're back with more This Week in Science.
Yeah, we're back. We are back. It's okay. It is time for that part of the show we like to call This Weekend. What has science done for me lately? What has it done? Oh, this wonderful letter this week. This letter makes me so happy. Dear Kiki, Justin, and Blair, good science to you. Firstly, I'd like to say thank you for making my favorite podcast and source of fun facts. You're welcome. Thank you for us being your favorite. That's awesome. Since discovering TWIS some months ago, I've been having such a great and interesting time going back through the archives of Twistery, apologies for that corniness, and having my mind blown repeatedly. I work a very menial job as a stewardess on a yacht and have felt my brain slowly turning to mush with lack of stimulation. TWIS keeps me thinking and curious and has inspired me to really try and figure out what to do with my life. I'm 23 and desperate to go to university to study a science of some sort, but I just can't pick. Between marine biology, astronomy, and geology, my mind just can't be made.
Do all of it. All of it.
Do all the things. That's right. Find the thread. Put them together. I think this is exoplanet studies, right? Anyway, congratulations on getting to 13,000 subscribers on YouTube. I know this because I persuaded my friend and coworker Charlene to hit the subscribe button and had the supreme satisfaction of watching 12,999 turn to 13,000.
No way. Oh, that is awesome.
Yeah. So what has science done for me lately? Jumbo coffee queen percolators and the process of extracting goodness from coffee beans in general. The sheer efficiency of putting the grounds in the filter, some water in the tank, and within minutes having 1.9 liters of energizing Arabica infused hot water. It's almost magic. It's science. But I could not work it without it. I could not work the long shifts and maintain sanity without it. Science also gives us pure drinking water on board. We use a reverse osmosis desalinator to turn the salt water of the ocean into clean fresh water, which we then treat with a UV filter and can drink. So no matter how long we're at sea, we always have plenty of water to drink. Thanks again for this kick-ass show. I really do love it and find you all so cool and inspiring. Best regards from the Ionian Islands and from your #1 Zimbabwean fan, Sarah Forfar. And she says, P.S. Blair, I recently saw a video of these cute little scorpions, or maybe they were tiny crabs, and they were all scurrying around on a piece of, I can't read this. I need to move my screen around. Oh, and they were all scurrying around on a piece of paper, and then someone would draw a circle around them in pen, and the little guys got super confused and seemed trapped in this little ring of ink. Can you explain what's happening here? No. Thanks, and have a fantastically scientific week.
So I can do some research and maybe be a little more scientific about this, but what I can tell you is I also saw this on the internet. It made the rounds. It is a relative to scorpions, but they're kind of almost tick-like, but they have these giant, basically like crab claws. They're great. They're so funny looking. But they are responding to the pens kind of like how if you put a square of tape on the ground, a cat will sit in it, right? So there's some sort of psychological process happening where they think it's a barrier, even though it's just pen. Again, I'm not sure exactly biologically if we know what's going on, if that's a trigger of something that happens to them in their environment. I could do some more digging and try to figure that out. But for whatever reason, yeah, the pen is making them think that there's a barrier. I don't know, Kiki, if you can find it, but it's maybe in the after show. I'll see if I can find it and I'll send it to you. But it's it's somebody who just has these little critters on a piece of paper and they draw a circle around it and they're like, uh-oh. And then they back up over it accidentally. And then they're like, how'd I get here?
I don't know how'd I get here. What? That's great. Yeah. I haven't seen it. I can't wait to see it. And maybe we can talk about it. Figure out what was actually happening. Sarah, thank you so much for writing. We appreciate your letter so much. Thank you for getting your friend Charlene to get us to 13,000 YouTube subscribers. That's wonderful. Everybody did that. It would double our numbers. If every one of our subscribers got one more person to subscribe, hello, we would be growing like gangbusters. That's how big an impact each of you has on us. Thank you for writing in. If any of you want to write in with what has science done for you lately, send me an email, kirsten@thisweekinscience.com. Put a message on our Facebook page. That's right, This Week in Science on Facebook. Send us a message and we will read it on the show. We want to keep this section of the show full of your letters, so keep writing in. And now we're going to keep talking about science. I'm getting sleepy. Oh. Do you guys feel like you are really well rested after, 6 hours of sleep?
Nope.
How many hours of sleep do you need to feel rested?
12.
So from the moment I hit the pillow until my alarm goes off, 8.5.
8.5. I'm at about 8.5, 9 myself. When I can make do on about a day of 6 hours of sleep, but
Yeah.
I am not the kind of person who can do that long term.
I've met people who continually and with the same energy they put into a normal day have done 4 hours a night.
Oh, yeah.
And
Yeah.
I don't understand it at all. It makes
I
no
had a
sense
professor who said not everyone needs 8 hours, and he got 4 hours sleep every night for his entire life. He swore, and he
Yeah.
thought it was fine.
And I've met people who are like this, who have like they go work out, they exercise. It's not like they're just conserving energy all day.
No.
They're going out and
They're-
being in the world like normal humans and having maybe more energy.
These people are mutants. They have a mutation or maybe a couple of mutations that have given them this ability to not need a lot of sleep. In 2009, researchers described a gene called DEC2. There's a mutation in it that led to a mother-daughter pair being very well rested on a short amount of sleep, 6 hours-ish. However, researchers now are publishing a new study in the journal Neuron, where they have studied another family who are all short sleepers, not just early birds, but just short sleepers. And they found another mutation, which is found in only about, they think the prevalence is only about 4 in every 100,000 people. So it's very rare. The mutation affects a gene called ADRB1, ADRB1. And this encodes for a receptor for noradrenaline. You guys familiar with noradrenaline?
Mm-mm.
No,
noradrenaline. Adrenaline. Does that ring a bell?
Mm-hmm.
Right.
Nora is adrenaline.
So basically, it's basically adrenaline. This is this signaling molecule in the brain, in the body. Noradrenaline. It's a stimulant in our brains. It wakes you up. And they looked at mice that have these neurons in this the brain stem and that have this receptor all over it, and they manipulated the receptor, so stimulating the ADRB1 brainstem neurons in the mice woke up the mice if they were in a very deep sleep. And from this study, identifying mutation in the family and also seeing how it works in mice and that they could potentially manipulate it to make the mice not need as much sleep, that this mutation is basically making these brainstem neurons more active. And if they're more active, then, and this is the part of the brainstem, this is the deep brain that makes everything else go. And so if those neurons are active and not needing sleep, then the rest of the brain and the body are going to follow suit. They want to do future studies to figure out whether or not they can recreate this mutation with drugs. So, for instance, make soldiers need less sleep, or shift workers need less sleep, or
what was the name of that movie? Limitless, right?
Right, Limitless. Yeah, maybe it could go the other direction though and help treat sleep disorders. So,
okay,
maybe this will give a clue in that direction.
I have a question.
Yes,
we need sleep. Right? Like biologically, that's what rebuilds telomeres. We can heal, right? So is there a potential negative impact to being a person who just generally sleeps less? Do these people not live as long? Do these people not heal as quickly? Or is there really no reason that we sleep as long as we do?
Yeah, I don't know that there is. I mean, we have this general idea that it's good for us, that we need it. And yeah, we do. People become basically drunk. You know, you have similar deficits to alcohol when you don't sleep. And you can die if you don't sleep. So it's important, but at the same time, the extent of its importance, whether or not it actually leads to longevity or not, we don't know that. And the evidence, as far as I recall at this moment, I don't think early birds, I think, are likely to live a long time. They're likely to live longer, I think. Am I remembering this correctly? I don't know. I'm going to have to look into that a bit.
Like shift work can impact your life expectancy too, right? So,
right, but that's circadian rhythms and being off rhythm,
right? But I thought that was because the quality of sleep was different because you're not supposed to be asleep. So this is kind of down to the initial question of if you don't need as much sleep and you're a shift worker and you can suddenly make yourself okay on 4 hours sleep, well, that reverse some of those impacts because you're not getting as many quality hours as this person who's laying down for the same amount of hours, but at the right time of day. So
right.
I think really the linchpin here for me is whether there's any negative impact of sleeping less if you have this mutation.
Well, I guess that it's a little bit of a troubleshooter if there is, and if this population of short sleepers could be just studied through metadata or something that's already out there. But it might be interesting to know if there isn't any real health deficiencies from a 4-hour sleeper. Really, maybe all the healing time could be done in less than 4 hours. Maybe it's done in 2 hours. Maybe it takes place right away, and it doesn't really require.
Or maybe it's kind of like rendering cycles where maybe it like goes over it once and then doing it again helps it be higher resolution. So doing it once is fine. Sleeping another sleep cycle just sets everything a little bit more solidly. I don't know.
This is blowing my mind because biologically, animals need sleep. And if there was a mutation that occurred naturally that allowed you to sleep less and not have any negative impacts, I feel like that would have rippled through the animal kingdom, except with everything except for maybe, I guess, apex predators. I don't know, which technically I guess we are, which might explain some of that. But there are animals that are potential prey animals that still sleep a lot of hours. And it might benefit them to be awake and to be grazing or to be actively aware of their story. Like, do you understand? I'm so confused.
So one of the things I could throw out there, at least that would be beneficial for that would be less beneficial for the rest of the animal kingdom, but can possibly work that way with humans, is that we can turn this daytime on anytime we want. Most of the animal kingdom, there's going to be a period of time when their eyes aren't adjusted for the world that they're in, or that the prey that they would be going after may not be out there. So there's a lot of, I think, more, why fix the thing if it ain't broke? It's going on in the animal kingdom. They don't have shift work like we do. They have pretty regular schedules.
Yeah, I don't know. I just feel like you would see more of this, even just in humans. Like, there's a benefit to being awake more.
Okay, so here's the next place to look. Here's the next place to look. You go to Arctic regions and see what sleep cycles are like during the 24-hour sun times and see how those might be different. That would be kind of fun to take a look at.
Mm-hmm. Right, when the sun is out all the time, right? Yeah. That would be interesting. Well, I mean, the interesting aspect is these short sleepers who have the mutations, they are just fine. So they're not feeling a detriment. They're not. There's no detriment to them.
No, I feel a little bit cheated not having this gene.
Yeah.
I know. I want it.
4 hours of my life every day. There's stuff I could be doing with this time.
Yeah, I just found a statistic that it's only about 1% of the world's population are these short sleepers who need, say, less than 6 hours of sleep a night.
I mean, the conversation we were just having about the nudibranchs was all about trade-offs, right? So
right.
it's
the trade-off?
So hard for me to think that there is no trade-off for this. Congratulations, you just don't have to sleep as much. End of story. I feel like there's something. I want more studies. I would love it to be true, and I want that pill to quote the film. But I want more studies because I'm very curious. I really feel like there's something else here.
There might be. There might be. Well, there are other things out there, other planets that we might want to look at, for instance. And when we look at them, we're looking for life, really, right? We want to know whether, or the potential for habitability. And we've talked before about kind of that, the gases in the atmosphere and what we're looking at around these planets. But we so far can only really see visible light, and visible light doesn't give us, or what we can see so far doesn't give us the exact spectrum to determine a lot of the chemical makeup, the chemistry in the atmospheres surrounding these planets. And that's something that's going to be really an essential part of determining whether or not the atmosphere is suitable for life. So some researchers at McGill University, this is an undergraduate with her advisor writing on her undergraduate thesis, put this project together and published in the monthly notices of the Royal Astronomical Society just today. They have modeled, based on satellite data, the SciSat, SCISAT satellite data, they've constructed an infrared fingerprint for the Earth. So this is a satellite, a Canadian satellite, that looks down on our planet in the infrared to be able to look at particular biosignatures for various purposes. And they took the data, created this fingerprint with the specific idea that nobody yet had created this image. Nobody had looked using infrared at the Earth and put it together and said, this is what we're going to look like to aliens, really. And so now they've published it, and now we have a biosignature, a fingerprint that we can try and match other exoplanets to Earth.
That's a very clever idea.
Yeah, it's a pretty neat idea. So the this SciSat satellite is interesting because it does kind of look at the Earth's atmosphere. It's looking mostly, it was started to, it was created, not started, it was created to help us understand what was going on in the ozone layer. And so it was looking at our planet as the Earth passes in front of the sun. So really it was looking at a transit of the Earth in front of the sun, which is exactly how we look for exoplanets. We just don't yet have an infrared telescope out there to make these observations, these transit observations on other planets. But dun, 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 the James Webb Space Telescope, which is going to be an infrared telescope with a launch date of 2021, which is finally completed. They built it. It's all in one piece now. This is really good news. I mean, I've been on the doubter side of whether the James Webb Telescope is ever going to end up out there. But according to news out this week, yes, indeed, we've built it. The next step will be launching it. But once it gets out there, these researchers also modeled the TRAPPIST-1 system. And this is a system, a red dwarf star, which is kind of a
close planets.
Yeah. It's got some really close-in planets that they pass in front of their star fairly regularly, which would make viewing them pretty easy. And they also, additionally, being a red dwarf, it's not really bright. So the brightness of the star doesn't, it's not so bright that it outweighs that dimming from the transits of the planets that are going around it. And so it's a great system to model. And so they modeled that system in this particular study and showed what they should be looking for on the various planets that are orbiting around the TRAPPIST-1 red dwarf star, and how we should be looking for it. So modeled it in the near infrared and also medium infrared wavelengths to determine how we can look at the planets to figure out whether they have oxygen, hydrogen, nitrogen, what is in the atmosphere. So Pretty exciting.
Right.
Fingerprint for Earth. We now have an infrared one.
Nice.
It's pretty cool. Tell me a story, Justin.
Okay, so this is another Toxoplasma Gondii story. The baby blinding, suicide, and schizophrenia triggering one of the leading causes of death by foodborne illness. Single-celled parasite that only reproduces in cats. And it is at it again. This time, it's killing sea otters.
It's been killing sea otters, hasn't it?
Well, yes, it has. This is actually something that did start back in the 90s, at least the study did, into the death associated with T. gondii and sea otters in the wild southern sea otters of California. So there's lots of infection in this population with Toxoplasma gondii. But it is only, according to what I was reading, the infection is fatal for only a fraction of sea otters with the parasite, which is not a lot if you assume a fraction to be small.
Right. Fractions come in many sizes.
Yeah, you can have a fraction of any fraction itself is just a big
So they just mean not all of them.
Yes. Yes.
It's only some.
But if they had said one, you would assume, oh, well, unlucky otter. But no, that would have been all of them based on anyway. So according to this was a examination of sea otter carcasses between 1998 and 2001. T. gondii was determined to be the primary cause of death for 17% of the otters, and it contributed to mortality in another 12%. So pretty significant fraction of the population. But what was interesting is the T. gondii infection is much broader than that. But of course, not all T. gondii strains are equal. There are different varieties. This is the study came out of University of California at Davis, and it identifies the parasite-specific strains that were linked to the deaths. And it turns out this study was pretty clever because they were also tracking felines on land. And they managed to trace the strain of T. gondii that was killing these sea otters to feral domestic cats and bobcats in a nearby watershed.
So this is directly related to the question I asked on the show last week, whether it's just domestic cats or if wild cats are affected. So it sounds like, indeed, yes. Wild
it usually said that it reproduces in felines.
Yeah.
So it's
but colloquially, I never really know if they mean like all felines or just,
Yeah, it's got to be all the cats. Well, I don't know. It'd be interesting. I mean, you could find out really quick whether or not they have the gene to produce the enzyme that breaks down the linoleic acid. Right?
Well, I remember a number of years ago when we were first talking about sea otters with Toxo, there was a hypothesis that it was coming through flushable kitty litter into the water column.
Yeah.
Yep.
So this is sounding like, at least in Southern California, sounds it's coming from cats in the area.
Well, so the strain that's killing them is different. So it looks like what I guess we could still assume is that it could still be flushed kitty litter from released sewage water that is infecting the majority of them. But in this but it's the deadly strain seems to be coming from the watershed and is being because they could actually find it there in the feral cats and the bobcats. So it's really interesting because they linked on land to what's going on in the waters. One of the other interesting things I think about this study is they point out that sea otters are very well studied in the marine life off the coast. There's only this sort of general knowledge of Toxoplasma gondii effects on other marine life. So it's not that it's just attacking sea otters. It is that sea otters are very well studied. And that's sort of how they were able to detect and put this. There was previous research that showed that up to 70% of stranded sea otters were infected with T. gondii. I think that might be the study that sort of got this interest kicked off and got the UC Davis folks involved in doing this further investigation.
That is just we've known about Toxoplasma and sea otters for years, and I never was like, oh, what other animals is it affecting? Where else is it affecting? It was just scientists discovered it in sea otters, so that's the species that they discovered. I thought it was a specialized kind of thing, not just that sea otters were very studied, and so that's why they found it in them.
Yeah.
They just hadn't looked at other animals, maybe.
Yes.
So if you think about it, you have felines, you have rodents, you have now mustelids, you have weasel family, and now you also have this idea that it affects humans as well, primates.
Oh no, it's very well known that it affects humans. It's basically anything that's warm-blooded. They know,
right? So it's this cross taxonomic. Huge groups, right? So it's not even like, oh, it's just carnivores, because you have rodents in there. So it has some sort of impact across all these different kinds of mammals. So it does kind of make sense that just if you look hard enough, you might find it in some more. It's just that, if you find a dead or ailing sea otter, it's going to get tested. Absolutely, because they're so well protected. Yeah.
But what about other animals? They're probably not tested, right?
Yeah, and so
how many?
It's because the more that we've been talking about this. For the entire length of the show's existed.
Yeah.
At this point, and there's never like, oh, Toxoplasma gondii found to be helping salmon. Yeah. And their salmon runs. Now, there's nothing that we're getting that's like really great news about.
You could even wonder, is it affecting deer? Are deer easier caught by mountain lions because of toxoplasmosis?
They become attracted to the scent of cougar pee. Yeah.
There's no way to know until you start testing it. It could be everywhere.
Mm-hmm. You know it's everywhere. What? You're going to say something?
Oh, well, yeah. Anywhere that there's cats, and there are probably 100 million cats or more in this country. And apparently it's not just here. It's everywhere. It's around the world.
Yep. Affecting sea otters around the world, maybe. But you know what's going to affect us right now?
Around the world.
Yeah, maybe around the world.
Around the world.
Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh my goodness. Well, if you've ever been to an airport, which I imagine many of you have, you may look out on the tarmac and see these individuals who are flashing their lights, helping direct the planes in, and they have big, fat noise cancelers on, trying to save their precious ears. Well, you know who don't have those big headphones to block out airplane noise? Just wildlife in the area, especially
everyone
birds.
Else.
Yeah. So this is a study out of Manchester Metropolitan University and the Institute of Biology, Leiden. And they have looked at the impact of anthropogenic noise on birds, specifically around airports. Now, earlier studies looking at birds like great tits and chiffchaffs.
Chiffchaff.
Say that a million times. It just brings a smile to your face every time. Chiffchaff. And many other bird species have found that when they are in urban areas, they have a noise-dependent shift in song frequency. So when they're in really loud urban areas, their song frequency will actually go higher. So they're trying to get over this low-level kind of hum of white noise of urban development. So their songs will go higher in pitch. And so that would in theory, help them be heard over the low frequency noises of traffic. Aircrafts are different. They are so loud and the noise is so wide in frequencies that it actually covers all frequencies of sound that we know about in terms of noise, right? So we can hear, that birds can hear. It covers all of the frequencies. So there's no benefit to them to going in higher in frequency because it's across all the frequencies, right? So this new study looking at specifically chiff-chaffs, it sounds like they sing at lower frequencies when they're near noisy runways. But they think that the reason for this, because remember, this covers all the frequencies, right? Is actually they think that that this is because they're going deaf. So just like, there's the very high pitched sounds that younger people can hear. And then as you get older, you can hear less and less of those. So that's what they think is happening here, that these birds are going deaf. So their songs are actually going lower. And so meanwhile, something else is also happening at British airports, and that is that birds are getting more aggressive. So there's a couple ideas why this might
hear
be happening.
Each other.
Yes. So the first actual hypothesis, the first hypothesis that they have, these researchers have about why they might be more aggressive, is actually that it's just noise-induced physiological stress. They are just stressed out and they're on edge because it's loud and it's right. Or it could be from disturbed communication. They just are having trouble hearing each other. I think that just when you're deaf or you're going deaf, but you used to be able to hear, you're probably more jumpy, right? If somebody can sneak up on you and poke you on your shoulder, you're going to, where'd you come from, right? So if you're a bird, it makes sense to me that you would be more aggressive.
But there's the other side of it, which is that birds use their calls to set out their territory. If you can hear me, you're probably too close to my territory. And so if they're no longer able to hear each other's calls, maybe at distance, they're getting closer together before they do come across each other. And by then, it's a situation of each bird thinking the other is on their territory. And so that's going to escalate quickly as opposed to just the call and response. Oh, okay, you're there. I'm over here. You're there. I'm over here.
Yeah. Well, and absolutely that. So in most kind of competitive displays in the animal kingdom, be it male lions competing for the head of a pride, or it's a couple of deer fighting over a specific territory with big antlers, with big racks. Those situations, they call them escalating behaviors, right? They kind of size each other up, get closer and closer, but in most cases don't ever make physical contact because it is so risky and energetically expensive. And this is exactly the same. They use their songs to identify their space. They will get closer and closer, but physical fights don't often actually manifest when these birds are trying to stake out their territories.
When they can signal each other with their calls.
Yeah, so they can signal each other, they can hear each other coming, all this kind of stuff. But the second that they can't hear each other very well or anticipate each other's movements, that becomes a problem. So, of course, this is a worldwide concern. There are airports everywhere. There are lots of animals that live near airports. In Manchester Airport, where they were doing this study, they they think there's around 16,000 individuals from 100 different species experiencing aircraft-related hearing loss. And so this could have, even just at this one airport, that's a pretty big impact. So knowing that, there's a lot of different thoughts about where to go from here. I couldn't help but think about the fact that there's been lots of conversation about the potential for electric airplanes. And even if you just found a way to use some sort of electric power during takeoff, you could greatly reduce the amount of jet engine noise.
Right. Any kind of noise reduction would be helpful. There's also the, hey, birds, maybe you should move someplace else.
There is that too, which is a very interesting point. Why are they hanging out?
Why are they hanging out?
I like the big metal birds.
The big metal birds keep the hawks away.
Yeah. So I will move from what is potentially a very unintelligent bird choice to a story about very intelligent bird choices. So I had to, of course, bring an amazing story about New Caledonian crows, our buddies that make tools, that store tools, that design tools, that can pick the best tool for an opportunity. They are so good at it. They are so smart. And a new study, this is coming out of Harvard, tells us that they might actually really enjoy making tools, using tools, and solving puzzles. So this is a study looking at optimism in crows before and after using tools. And the lead researcher says, just the same way we enjoy something like solving a crossword, crows actually enjoyed using tools. They get satisfaction out of doing things they're good at, have trained for their whole lives for, and that they use frequently. So this sounds pretty outrageous that we'd be able to make this claim. So I want to take a step back and explain how they came to this conclusion. So the New Caledonian crows are super well known for their tool use, but they wanted to see how using tools made them feel. So what they did is in a lab, they trained crows to identify where different areas of the room would have larger rewards and smaller rewards. So they were particularly looking at one table, and on the left side of the table, if there was a box on the left side and the crow opened it, there would be a large reward, 3 pieces of meat. If it was on the right side of the table and they opened it, there would be a small scrap of meat, a far smaller reward. So they trained these crows to know the difference between right side, bad reward, left side, jackpot, right? And then they slowly moved that box towards the center. And eventually it was dead center in the table. And so that was measured as their optimism. Glass half full, glass half empty. If the bird quickly came to investigate the box, they were optimistic that there was a large reward in there. If they waited or didn't visit at all, they were pessimistic. There's probably nothing good in there anyway. So then they wanted to see.
Go
ahead.
I'm picturing the nether category was probably the outlier, which was the wishful thinking one that went and nudged it to the left or the right before opening it. Went and nudged it a little bit to the right side of the table. And
like
then
if I put it over there,
then
it'll magically have more. Yeah. Well, remember, crows also know object permanence. So they know that whatever's in there is in there. So interesting to bring that up. So then to test how this is related to tool use, they were put through a series of tests over a number of days, one in which they had to use a tool to get a piece of meat from a box. This is a different box. Okay. And another in which the meat was they could just grab it. So they did find that they were in a quote unquote good mood as a result of using the tools. But then they thought, okay, what if it wasn't them enjoying the tool use, but it was them working harder for food and therefore being more hungry, or that being more fun, or who knows, but maybe it was just working harder for the food. So then they added additional conditions. In one, the meat was on the table, no effort at all, just on a plate. And in another condition, they dispersed it around the enclosure in 4 different corners. So they had to fly around to get to the meat. So it was still more effort, but they didn't need to use a tool to get it. And they found that following tool use, the birds were quicker to approach the ambiguous box and much less enthusiastic after the effortful test as compared to the easy test. So it wasn't the effort, it was the tool use. They don't like tool use just because it's difficult. They like something specifically about it. So it would appear that they are enjoying it. And this is directly related to something that we talk about all the time when we talk about having animals in zoos and aquariums and science museums in captive environments, is enrichment. Right? Is trying to get them to do natural behaviors that they would do in the wild. So it's not just food on a plate. It's getting them to, if they're a vulture, stick their head into a box with a small hole in it to get their food out. Or if they're an animal that digs, putting it in something where they have to tear it apart to get to it. Right? So this is enrichment. This is encouraging natural behaviors to get to the food, to make it harder so that it's not just, oh, I ate and I'm done and I'm bored, but this is a whole nother side of it. Maybe it's part of their mental health. Maybe it's part of their, it's hard to say happiness or enjoyment, but something related to that. Maybe they're more content using their natural behaviors. It's pretty interesting.
It is interesting. I mean, I would love to know, the impact or how this works in species of different levels of intelligence.
Yeah,
I mean, not with necessarily the tool use, but in terms of enrichment. And could we determine whether generalized enrichment makes animals generally more optimistic about
well,
stuff?
we know that it reduces stereotypical behaviors. It reduces pacing. It reduces over grooming. So we know from a lot of behavioral studies in captivity that that enrichment generally helps the mental health of those animals. So we know that. So then, the question is, do they enjoy it? Does it create contentment? Or is it just giving them stuff to do so that they're not focused on, being bored?
Right. I mean, when an animal has stuff to do, they're less likely to develop stereotypical behaviors like feather pulling or hair pulling, or, so they give themselves bald patches and, these terrible things that are similar to human mental disorders. Animals can display symptoms of anxiety, of depression, it's interesting. This is similar to a study that we talked about a month or two back, but this idea of optimism and pessimism in animals, I'm still not enjoying the word choice.
Right. Yeah. Yeah. It could simply just be more curious. Would also be
engagement.
Engaged.
Yeah, more engaged. Yeah. Exploratory. There's lots of other words that you could use. I agree that might be more descriptive of what they observed. One of I didn't close it yet. One of the quotes from the lead researcher that actually, I don't want to put anybody on blast here, but it irked me a little bit, was that, quote, "I think we tend to under-anthropomorphize animals, especially really intelligent animals. It's not that they are machines and we are feeling beings. Clearly, animals also have emotional reactions and moods." So yes, animals have emotions. Yes, they have moods. But this idea that we under-anthropomorphize animals, I spent a lot of time on this show arguing the exact opposite of that. Animals are not humans, or wild animals are not humans. We are animals, but we are a different kind of animal. There are things that we think are unique to us that are not unique to us, but also imposing human experience onto a wild animal is very questionable. So I agree with you. I think the terminology could be better, but what we're seeing is that there is a potential increase in mental health based on mental stimulation, kind of.
Yeah.
So
there's a change in affect for sure.
Yeah.
They're more activated. They're more prone to continue the tasks that they're being given if they're in a quick manner.
Yeah, one of my favorite studies that I learned about when I was a carnivore keeper actually was on fishing cats. These little cats I think they're from Southeast Asia. that are one of the only cats that can swim, and they'll jump down and fish. And when you feed them live fish in a zoo, if you feed them a live fish every single day, you would think, right? Like, this is a great experience for them. Give it to them every day if you can. It actually, their behavior peaks for a couple weeks and then severely drops, and they start to ignore the fish. If you feed them on a schedule, if you give them live fish every Monday, Wednesday, Friday, exact same thing, peaks and drops off. But if you feed them at randomly kind of selected days, their behavior peaks and stays up because they don't know when to anticipate the fish. So they will go swim and they will look for fish, and there may or may not be fish in the pool. And that more directly replicates how things work in the wild, right? You don't always get food. But I think that this is exactly the point, right? Is that it's making them use their brain for what their brain is for. What has it evolved to do? And we're getting them to do that.
To deal with surprises, to deal with the unexpected things that occur in the environment. Yes, that's what the brain is tuned for. And when everything is the same all the time, yeah, any animal might go a little batty. Yeah. Moving on from crows and puzzles, human diet, that's another puzzle that's always interesting. I'm fascinated with the idea of caloric restriction as a way to increase health and increase longevity. And there's a new method of caloric restriction that's called intermittent fasting. We've discussed it on the show previously, where instead of just decreasing your calorie intake, say by 25% every day, all the time, you just don't eat for one day out of 7 or whatever schedule you end up subscribing to. And there are many different ways to choose this intermittent fasting and how you will eat. A new study has just come out, published in Cell Metabolism. Researchers have looked at the difference between just caloric restriction and alternate day fasting. And in this strict regimen, what you do is you fast for 36 hours, and then you get to eat whatever you want for 12 hours. Whatever you want. Anything. They found the 2 separate studies in this. One, they looked at 60 people over 6 months and found that it was similar to what had been seen in caloric restriction trials, where that's just diet, right? You just reduce your calories. They found that the people in this ADF, this alternate day fasting regimen, were able to actually stick to it better, and they had similar health benefits. And then they did another trial where they did a random control where people were put either in the alternate day fasting randomly or in a control group for 4 weeks, and they compared them directly and found that there were some very definite increases in metabolic factors that were beneficial. And they say that what ends up happening with alternate day fasting is that people end up over time eating less calories than they would have, fewer calories than they would have had they been only on caloric restriction. Because of the 36 hours of not eating, even though you can eat whatever you want in those 12 hours, most people do not replace all the calories that were lost from that 36 hours of fasting. And so in all, they end up coming out below what would have been, what would have occurred with just dieting, caloric restriction. And people are supposed to be able to stick to it more easily. I don't know if I would. The idea of 36 hours of not eating is it's a little scary to me. And doing that, 36 hours diet, 12 hours eat what I want, and just doing that over and over and over and over again. I don't know about. I like to fast.
It does explain why poor people are always so healthy.
No.
So I kind of do this diet once in a while, just sort of accidentally. Like it just,
You're going to live forever.
I push off meals, but yeah, I don't know. I don't know that it's, I know it's not for Blair.
I'd lose friendships. I might lose my job. I get so angry.
Yeah, when I don't eat, there's the low blood sugar factor that it's not so fun to be around, right? I would probably hurt somebody. It wouldn't go over very well. Yeah. But anyway, new ways of looking at dieting. We'll see if you would like. It's a new thing. It works. People say it's really good. The science is good. And this big news, really exciting. Researchers have remade computer chips just like they were in the 1980s, but not out of silicon, out of carbon nanotubes. This is the first time that engineers have actually created. This has been an idea for a long time. Researchers have been wanting to make chips, computer chips, out of carbon nanotubes. But for various reasons, technical hurdles, they have been unable to do so until now. And the researchers now were able to create this carbon nanotube chip using, there's some neat methods involved. Anyway, we'll put a link on the website. I want to get through the story fairly quickly. But the exciting thing is not that it performs as well as a 1980s silicon chip, which was pretty slow. Computing back in the 80s wasn't much. But what is exciting is that we finally have gotten to this point. Silicon is reaching its limits. The silicon chip is at some point going to max out, and we are going to exceed the energetic capacity, the amount of current that can flow between a gate, between in these circuits is going to be maxed out. We've gotten tinier and tinier and with more and more stuff packed in. Carbon nanotubes have the potential of going even stronger and with more computing power. So,
how much more computing power do we really need? How much smaller do the devices
give me all the computing power, all of it.
I mean,
I want to stream to 6 different channels at once.
Yeah, for personal electronics, aren't we there? Just stop. But for research purposes, I can see some fantastic benefits.
For lots of personal stuff, there's all sorts of reasons why things should be smaller. For devices that will help us with our health, little robots that could be working inside our bodies to keep us healthy. The new pacemakers that would be tiny at almost cellular sized. There are so many things. Neural implants. There are so many directions things could go with further miniaturization. Now, whether or not we want to go that way is another question. But the things that we can do or will be able to do will rely on having pretty large amounts of current being able to flow through very tiny, tiny channels. Yeah. Justin, how hot is it?
You're muted.
You're muted now.
Has anybody else noticed the... Can you see this?
Yeah, it's me.
I don't know why that's not on my end. Just so you know, there we go. Oh, yeah, that's me. There we go. That's even better. The story. It's getting hot in here, people. But not just in the United States. It turns out global warming is having an effect in Europe as well, which we don't hear as much about. Yeah.
Global, you mean, not just United States warming.
Well, I think, whenever they say global or the world this league or we're the world champion, it's usually just in the United States. Turns out, who knew, global warming is happening everywhere. This is increasing number of days of extreme heat, decreasing the number of days of extreme cold in Europe, according to a new study. So temperatures in Europe have hit record highs this summer, passing 114.8 degrees Fahrenheit in southern France, which is 46 Celsius.
That's insane.
Its research is in the journal geophysical research letters, and they find the number of summer days with extreme heat has tripled since 1950. Summers, therefore, obviously have become overall hotter. Number of winter days with extreme cold decreased in frequency by half. So not by as much, but still less extreme cold. Winters have become warmer overall, as well as, so summers are getting warmer, winters are getting warmer. A new study finds parts of Europe are warming faster than climate models had projected. This is quote voice for Ruth Lorenz, climate scientist at Swiss Federal Institute of Technology in Zurich, and the lead author of the new study. Even at this regional scale over Europe, we can see that these trends are much larger than we would expect from a natural variability. That's really a signal from climate change. It says extreme hot days, there's 3 times more of them than there was in 1950, and they're 4.14 degrees Fahrenheit, or 2.3 Celsius, hotter. Cold days have warmed by 3 Celsius, 5.4 degrees Fahrenheit on the averages. Those are on the averages. The hottest days and coldest nights were even more than their mean temperatures. So, yeah, it's getting hot out there, people, and it is turning out. I'm getting confirmation from the looks that my co-hosts were giving me. Global warming is actually taking place everywhere in the world. So.
It is. That it is. Around the world, global warming, the general term for anthropogenic climate change happening everywhere. Oh, Blair, you got another story?
Yes, it's actually related to climate change, so I'm so glad that you brought that up. Migration, animal migration. We've been studying it for a very long time. There's lots of question as to how the animals know where to go and when. Generally speaking, there are 2 camps of thought. One is that the animals use local cues within their vicinity to determine where to migrate. And the second is that they develop a memory of the landscape where they live and then use that information to guide them. So either the good food's over there, let's go that way, or that's where grandma told me to go. So University of Wyoming has a new piece of research looking at mule deer, and they found that mule deer navigate in spring and fall, mostly by using their knowledge of past migration routes and seasonal ranges. So in this case, this means that landscape memory is strong within a population. And so loss of migratory population numbers will destroy the herd's collective mental map of where to go. So the idea is here that if there's a reduction in the population size, there's less collective knowledge of where to go for migration. So it's kind of stored in the population as a whole. Which way do we go? I think we should all go that way. The other side to this is that there is an expectation as climate shifts or as habitats change from human impacts from deforestation and stuff like that, that animals will be able to figure out where to go next, or that if one area usually gets rain, but another area got rain instead this year, they'll go over there because that's where the food is. But if they're moving based on a mental map within their population, they might not catch on. So the more we know about what influences migration patterns, the better we can anticipate needs to guide them elsewhere, or what may or may not happen as a result of moving targets. Because one of the biggest problems that climate change brings to migrating species is a less predictable timeline and location of food.
Right. So it's that predictability that is, I love the concept of the community mental map in this situation. I hadn't really considered that before, but each individual has their own memory and may have experiences that would be beneficial in one situation or not in another. But overall, in the population, they can all influence each other and end up, it's kind of that the wisdom of the herd like literally.
Yep. Absolutely.
That's awesome. That is awesome. Well, I hope that the wisdom of the herd got you listening to TWIS in the first place. I hope that the wisdom of the herd has kept you listening and that you enjoyed this episode. We've come to the end of another episode, and it is time for me to say thank you to all the people, all you. I'm saying thank you to you for watching, you for listening. Thank you to Fada for helping with show notes and social media and our chat room. Gord McLeod, thank you for helping make that web chat Freenode chat room a good place to be. Identity 4, thank you for recording the show. And thank you to all our Patreon sponsors. I would like to say thank you now to those Patreon sponsors. Thank you to Paul Disney, Andrew Swanson, Richard Onemus, Craig Landon, Ed, Andy Gross, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill Kay, Bob Calder, Time Jumper 319, Eric Knapp, Richard Brian Condren, Dave Neighbor, Dan, Matt Bass, Darwin Hannon, Patrick Pecoraro, Ben Bignell, Gene Tellier, John Gridley, David Williams, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stephen Albaran, John Ratnaswamy, Dave Friedel, Gerald Myshak, Paul Ronovich, Sue Doster, Dave Wilkinson, Richard Porter, Noodles, Kevin Reardon, Christoph Zukrunarek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Jim Drapeau, Greg Riley, Sean Lamb, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you all for your support on Patreon. And if you would like to support us on Patreon with a monthly donation, you can find information at patreon.com/thisweekinscience or just click the Patreon link on TWIS.org. And remember that you can help us out just by telling your friends about TWIS. Get them to subscribe today. And on next week's show, we will be back yet again to talk about science news. We will be talking with a neuroethicist next week, talking about the ethics of various neuroscience policies in their involvement in the legal realm. So many things to talk about. The future of the brain and humanity. Maha! Yes. Once again, though, 8 p.m. Pacific time at twis.org/live. You can find us there, watch live, join the chat room, or you can watch later. We're on YouTube. We also have our wonderful podcast, which you can find at twis.org.
Thank you for enjoying the show.
For more information on anything you've heard here today, show notes will be available on our website. You know what it is. It's www.twis.org. While you're there, poke around, see what you can find, make comments on previous shows, start conversations with us, the hosts, or other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in your subject line. Otherwise, what will happen, Blair?
You'll be spam filtered into oblivion.
You can also hit us up on the Twitter where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover, address a suggestion for an interview haiku that comes to you tonight, please let us know.
We'll be back here again next week, and we hope to join us again for more great science news.
And if you have learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of Toxoplasma Gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.